Good day, ma'am. We are the group 2. And the title of our selected research is Hazard Analysis and Storage Studies on Tilapia. Saraketon Masambicus. Fish Fingers.
Chapter 1. Authors: Reggie S. Alejandro, Lorena S. Alejandro, Rosalie S. Benito. Objective: this study aims to identify possible hazard during tilapia means and surimi fish steak processing using established guidelines from the United States Department of Agriculture or USDA, Food and Drug Administration FDA, and Asian Canada Fisheries Post-Harvest and Technology Project. Second, establish a hazard assessment table which could be used in the preparation of HAC plan for tilapia means and surimi fish steaks processing. Monitor microbiological, sensory, and chemical change in a frozen tilapia means and surimi fish steaks over 3 months storage period. Background of the study. Researchers from the Bureau of Fisheries and Aquatic Resources, or Region 9 or BFAR, have developed tilapia fish fingers using mince and surimi from tilapia flesh mixed with shrimp, spices, and seasonings with fish finger were found to be highly acceptable to 100 consumers type penalties. To boost the marketability of the product, technical aspects like shell and food safety were established. Researchers conducted frozen storage studies and hazard analysis on the processing of the fish finger. The HACCP, or HAP, a food safety management system, is now required for many food processing plants in first world countries. The identification and risk assessment of the hazard in tilapia fish finger would help processors establish critical control points and measures to ensure food safety. The analysis of biological hazard is limited to Salmonella, fecal coliform, and Staphylococcus aureus.
I'm Caren Bayawa. Let us discuss the chapter 2. First, the hazard analysis concept. So, hazard analysis and critical control point is a food safety management system which concentrates prevention strategies to know hazard and the risk of them occurring of specific points in food chain. It is becoming a necessity for all food processors. So the second is applications of hazard analysis and critical control point in seafood. So microbiology analysis of seafood and their production plays a key role in assessing the safety, quality, health, and compliance with regulatory specifications. Traditional microbiological testing methods are often shown laboratories provide only related data a finished product. So, it is a cost-effective system that target resources to critical areas of processing. So, reducing the risk of manufacturing and solving unsafe product. Okay, so let us talk about the fish finger. So, the fish finger are the fried in vegetables oil, packed into wax wrap cartons to reduce moisture loss during storage and quick ground in plate freezer. So, most fish fingers contain 57 to 65% water and 10 to 14% protein. So, 6 to 105 fat and 1 to 2% ash. So, correspondingly to fish contents of about 50 to 70%, some authorities applies a minimum standard of 65% of fish. So, the changes in quality of frozen fish. Once the fish is frozen, bacterial spoilage caused by the exogenous enemies of the bacteria causes and autolytic changes caused by the breakdown of the chemical constituent of the fish by the enteric tend to be relatively show and contribute later to loss of quality. So, the major causes of quality loss during town storage or showing of dehydrations, drip loss, protein dehydrations and discolorations, and cold stored odor. So, the biology of tilapia. The tilapia fish utilization has been found ideal in the Philippines, most especially here in Zamboanga. So, the most common fishes found in the peninsula are bangos, or the scientific name Chanus janus. So, tilapia is the one of the freshwater species abundantly available, but it has no central value of a freshwater. And tilapia was detected to the preparations of men's for several reasons. So tilapia was recently been classified into three genera. One is a distinguished characteristic between the genera is the type of care of the parents provide to their young. And second is the of genera, so Saroteriton and Oreocorimis, the parents will will incubate and protect the young and their mouth. The last is particular the Ariomachis and plasticing distinguished by material mouth, bringing their parental care of the young and performed by the female.
Materials and methods. Means and surimi production. The tilapia for the production of minced fish and surimi were harvested from the ZSCMST Aquaculture Fish Pond. For the preparation of the minced fish, 10 kilo of tilapia were weighed and washed thoroughly with chilled water to remove dirt such as mud, sand, and slime on the scales of the fish. The washed fish were drained and then scales were removed using a kitchen knife. Fish were beheaded and gutted by cutting off the heads and removing the internal organs. The gutted fish was again washed thoroughly in chilled water to remove surface contamination, loose scales, blood, and pieces of gut, and then drained in the colander. The drained fish meat was weighed twice. The weight are recorded. The fish meat was then chopped a pre-cooled silent cutter and then weighed again. Then was packed in the polyethylene box and stored in a freezer at 1 Celsius. It is important for us to know these steps to prevent spoilage and maintain our food safety. Hazard analysis. Hazards associated with minced fish and surimi production were identified through 1. A thorough review of literature in surimi and minced fish technology, chemistry, and microbiology. 2. A thorough observation of the steps in production. 3. Microbiological analysis of the production to prevent spoilage and maintain food safety. 4. Microbiological analysis of swabs taken from workers, handlers, equipments, and utensils. Proximate analysis. The proximate composition of both minced and surimi fish sticks were determined using standard methods. Fat and proteins were determined at laboratory while ash, moisture, and crab's fiber content were analyzed at ZSCMST Microbiology Laboratory. Storage studies. Products were stored at 1 Celsius for 3 months. Samples were analyzed monthly for sensory score. Sensory evaluation. Sensory evaluation was performed by untrained finalists utilizing a 5-point hedonic scale for 3 months starting on each 0 month of storage. Sensory evaluations were judged according to texture, flavor, and general acceptability. The total volatile-based nitrogen, or TVB-N. The TVB-N of the samples was determined using standard methods by analysis of the BPAR Region 9 laboratory.
The BFAR standard required for fish and fishery products was BVBN 20 milligram nitrogen and 100 sample. Total plate count or TPC. TPC was obtained by plating samples dilution at 23 to 27 Celsius for 3 days. TPC were done at the ZSCMS3 microbiology laboratory monthly for 3 months. So, pH. The pH of the sample was analyzed by BFAR Region 9. Sampling were done monthly within its 3 months of storage.
So hello again, ma'am. I am Patricia and I'm discussing the chapter 4, results and discussions. Hazard analysis. So hazard analysis and risk assessment of fish stick starts with from the time raw materials are received and continues until preparation, processing, making, and storing. So seafoods, ay yung mga nabuborn na pathogens in seafoods is Staphylococcus aureus, Beribio species, and Clostridium botulinum. In these studies, yung mga handlers is analyze the Staphylococcus aureus and fecal coliform knives, chopping boards, water, and raw fish. Sa salmonella naman is butter and the fish sticks, yung mga inanalyze nila. And yung results ng pag-analyze nila is negative siya. Wala yung microbial na nanggagaling sa mga ano na yon. And sa using the pertinent data from USDA's or United States Department of Agriculture's from time temperature guidance for controlling pathogens, growth, and toxin formation sa seafood. So, table point 4.3, time temperature guidance for controlling pathogen growth and toxin formation sa seafoods. So, yung mga potential hazards na makikita mo natin doon or microbes is yung Clostridium botulinum, Salmonella, Vibrio, mga dalawang klase ng Vibrio, and Escherichia coli, yung mga makikita natin doon. Yung depende din sa yung type exposure nila is depende din sa kung gaano ka taas or kababa yung temperature ng pag-store nila. So, sa case na study na to is only 20 minutes siya in-store sa ice chest and with temperature of 1 to 2 Celsius. So, yung ano niya, hazard na ano niya is low. Hindi siya na ano kaagad ng hindi siya gaano na ano ng microbes. So during mincing and grinding, handlers wear gloves to prevent contamination to the seafoods para hindi masira yung in the study na ano. Kasi yung skin natin, yung throat, and yung nostrils is carry siya ng microbes kagaya ng Staphylococcus aureus. So masama talaga yon sa studies na ginagawa mo pag na-contaminate na siya. Buttering steps of fish sticks was considered hazard kasi yung pathogens na presence doon sa butter may be transferred sa or mag-grow during the lag period. Tapos yung Staphylococcus aureus naman, considered din siya na hazards in a step that pag-fry ng isda sa after extra toxins and resistance of 60 Celsius for 16 hours heating. So sa case na to is the 3 lag period between buttering and flash frying would have been enough for contaminating Staphylococcus from the contact surface to produce toxin. Sa microbial changes, TFC values of fish fingers from both mince and surimi remain fairly constant during sa storage ng isda. So sa ano naman, expected since microbial growth inhabit at very low temperature, pero ano siya, mahina siya na maka-grow. So, sa value also remains below the maximum allowable limited for frozen breaded fish steaks. So, throughout the entire storage, indicates the fish sticks remains acceptable in terms of microbial quality until 3 months storage.
Appendix C. Salmonella test. The letter A, pre-enrichment. So, 225 ml of sterile buffered peptone water with 25 grams of the test sample. So, ensure true mixing if needed. So, homogenize the sample and transfer the homogenate to a sterile 500 ml flask. Incubate at 37 degrees Celsius from 18 to 24 hours. Letter B, selective enrichment. So, resuscitated culture and inoculate 1 ml into 10 ml of SBGS broth and 0.1 ml into 10 ml of RV broth. So, incubate SBGS broth at 37 degrees Celsius from 18 to 24 hours and RV broth at 42 degrees Celsius for 18 to 24 hours. Inoculate control cultures into each broth. So letter C, planting onto selective agar media. So strict inoculate from selective enrichment broth onto XLD and BS agar plates. So inoculate at 37 degrees Celsius. Appendix B, coliform test. First, collect sample of water and food in sterile container. Next, for solid food sample, weigh 10 grams into 90 ml. Homogenize, prepare a 10 and 10 cereal dilution using peptone dilution fluid. So, for liquid product, water in dilution, sample may be used as 10 degree dilution in coliform MPN. Inoculate 1 ml volume from each of the dilution 10 to 10 to 10 to 0, 10 to negative 1, 10 to negative 2, 10 to negative 3, 10 to negative 4. So, into each of 3 tubes of lauryl tritose broth, inoculate positive and negative control tube. So, LT broth label very carefully. Next, incubate the 14 tube for 24 hours at 37 degrees Celsius. Then record gas formation in the Durham. Then record the result. Appendix D. Determination of moisture control. So, balance the infrared meter to 0. So, weigh the sample in grams and evenly spread it on the dish. So, put the dish on the infrared meter holder. Level the balance set lamp height to Mark 7 and turn on the moisture meter. So, dry until the scale readout stabilized, typically taking 30 to 45 minutes. Appendix E. Determination of ash content. So, weigh 4 to 6 grams of food into an ignited cold crucible. So, for high moisture, food evaporate to dryness, heat gently over a Bunsen burner until charged. So, transfer the crucible to a muffle furnace at 550 degrees Celsius for a while for a white or gray ash result.